So, good afternoon everyone, my name is Wang Yingwen. The bone broth is one of the major reasons that make the food taste delicious, because inside the bone, which called the bone marrow, is full of fat. This makes the soup become rich and flavorful after a long period of stew. So, it sounds great when we are talking about the food, right? But when we are discussing about our body composition, it is not happy to see too much fat accumulate in our bone marrow, because it will lead to lots of problems such as high fracture rate, impaired hematopoietic potential, and a disease called osteoporosis. So, why the fat tissue accumulate in our bone? To answer this question, we have to understand the bone marrow-derived mesenchymal stem cell first. The bone marrow MSC have ability to differentiate into osteoblasts, the bone cells, to maintain the bone integrity. However, the osteogenic capacity decreases with age, and they prefer to turn into adipogenic cells and let the red bone marrow become the yellow one. This is a defect mostly controlled by the microenvironment cues. One of the major superfamily ligands called TGF-beta is the most abundant cytokine in bone. In general, the prototype protein TGF-beta sustains the stimulus and promotes the proliferation of MSC via the SMAD-Q3 signaling, and therefore block the differentiation pathway. However, the other subfamily ligands called bone morphogenic protein has totally opposite biological function of TGF-beta. Its induced SMAD158 signaling is to promote MSC differentiation. So let's give me a thought that if we can distinguish the signaling pathway of these two inhibitory effects, then we can counteract the adipogenic capacity of BMP, but do not decrease its osteogenic potential. The answer is yes. We found that TGF-beta goes through the canonical SMAD2/3 signaling to inhibit adipogenesis. However, it kidnaps the SMAD1 to form a novel mixed SMAD complex with a specific co-repressors histone deacylase to inhibit osteogenesis. We therefore combine the TGF-beta and BMP with a small molecule inhibitor to block the biological function of the mixed SMAD complex, and found that the bone mass was significantly increased. So this result provides us a new strategy in therapeutic use for aging or obese individuals with adipocyte-enriched bone marrow, and thus reduce the risk of osteoporosis and the bone fracture. Thank you for your listening.